for homogenization of the tissues, plant samples can be used by pestle and mortar grinding or by using a bead build method. So the bead build method, you need purlin beads. This is the purlin beads. So we are going to collect the samples with the purlin beads filled with Eppendorf. So sample, take little bit amount of purlin beads. It's around 300 microgram. With I am going to handle 4 samples. With the 4 samples, 4 Eppendorf, I am going to fill with the 300 to 400 mg of beads. So all 4 vials were filled with purlin beads. So in the field, you have to go and take this purlin beads filled with Eppendorf. These Eppendorfs can be directly taken to the field and mark the sample, what sample you are going to take and which line of the sample which you are going to collect. We are going to isolate the DNA from both monocot and dicot plants. The monocot example is rice and dicot example is bougainvillea. So for collecting of the sample, we should not touch that leaf because we have the surface pathogens and it won't get contaminated. So just collect the samples like keeping inside of the plant and just close the lid. And it should be around 300 for Salk line method of DNA isolation. So I am going to take 3 plant for making that average. Actually growing and young leaves can be collected. After collection, immediately the sample has to be frozen in the liquid nitrogen. So I am going to take that liquid nitrogen in that vial. So the sample has to be frozen in the liquid nitrogen. After frozen, it becomes brittle. And brittle samples has to be directly taken to bead mill pulverizer or homogenizer. Yeah, the frozen sample is ready, and immediately keep inside the sample and close the lid, and I am going to keep for 5 seconds, and after 5 seconds completed, 2-3 cycle you can go for 5 seconds each. Then after bead mill or homogenized, you will have the sample is very clear powder. With that powder, you can proceed with the protocol as mentioned and add 300 microliters of Salk line extraction buffer. Pulverized liquid nitrogen sample is ready, and now you have to add 400 microliters of this is the Salk line extraction buffer. I am going to add 400 microliters of Salk line extraction buffer to it. And one more sample, this is a monocot and another one is dicot. So now sample is ready. and you have to just gently invert like this for 5 minutes. Then after 5 minutes, centrifuge 10,000 your sample inside, and then after setting, just rotate this knob to reach 10,000. After reaching 10,000, you set the time 5 minutes. After completing centrifugation, take the sample. Without disturbing, I've collected after centrifugation of 10,000 RPM 5 minutes, 300 microliters, and to which I'm going to add equal volume of cooled isopropyl alcohol to precipitate the DNA. And now I am going to take while adding isopropyl alcohol, you may have the thread-like structures of the DNA get precipitated. I'm going to gently invert. As you can see, the thread-like slimy layer is moving. Look at that. That is the DNA. And invert gently like this for 5 minutes at room temperature. Then again centrifuge 10,000 RPM for 5 minutes and collect the pellet. The pellet can be dried and it can be used for further use.